This is the first prep. There might only be two preps for this lecture. We're going to do some practice in the class with mapping by this method, but the lecture notes themselves won't take as long. We're going to finish up with bacterial genetics. We spent the first lecture talking just in general about the methods by which bacteria can exchange genes. We will not be talking about the viral transfer. I've decided, because when you look at co-transduction frequency, the likelihood that two genes are transferred together by a virus into a recipient bacterium, the method and the thinking is exactly the same as co-transformation frequency, and I don't think that the time we spend will enhance your understanding. It's just another name for almost the same thing. Instead, what I'll do is I'll talk just briefly at the end about multiple drug-resistant bacteria and a little bit about just, how that happens. What we're going to do today is follow up on the gene mapping in bacteria, which we started last lecture, and that was using co-transformation frequency to determine if genes are close together or far apart on the bacterial chromosome, studying them in pairs, and then interrupted mating experiments where we were able to determine, based on timed interruption of the conjugation process, how long it takes for a particular gene to be taken up from an HFR strain into a recipient, and how much time there is between taking up the first and then the second gene and then the third, and so on. So we get a sense of the relative order, and we get, if they're very close together, we can get that muddled, though if they're only 1 or 2 minutes apart, that's a little dicey. But we get a sense of the order and a reasonable idea of ones that are further apart from each other and ones that are close together. Today we'll move to using HFR mapping for a really precise, detailed look at how to map those genes, and the information we get from the experiment is much more reliable for future experiments, and it requires careful design. Badly designed bacterial genetics experiment is either completely uninterpretable or like massive amounts of work. If you design it well and you do, your interrupted mating experiment in advance, or use somebody else's data to properly design, then you'll have interpretable data and good quality information out of the experiment. Okay, so this is just a little review. The co-transformation frequency that I already mentioned. Are these two genes close together, or are they relatively far apart? And we can use HFR strains with the F factor to do interrupted mating. And so that allows us, we let conjugation begin, and then we stop the process every minute or every couple of minutes, and then test with lots of different replica plating to figure out the genotypes of all the colonies that result. And that helps us understand, approximately how far apart genes are and approximately what the order is. We can do that with a few genes at a time. And then we can use that information to study those genes, but to map them in more precise detail. Okay, and so this is how we start. So when we map genes the sort of very high resolution, the very precise way, we do it the same way we do with eukaryotes. We are basically counting the number of recombinant events that occurs between the genes, and divide by the total. The trick is, how do you know what the total is? Because you could plate, 1 million cells after conjugation, and only a certain number of them have been able to have had the opportunity to undergo mating. Only a few of them have taken up the HFR, some of the HFR DNA. In some cases, they may only have taken in a very small amount before the mating process got interrupted. In some cases, they may have taken in lots of DNA. And so we're really, we're dealing with a high number of variables, and we really don't want to deal with that many variables when we're trying to get good interpretable data, right? Data that we can rely on so that if we did the experiment again the next day, we'd get about the same results, right? You want something you can depend on. And so in this case, what we do is we decide, okay, let's say we decide we want to map the B and the C genes, and we relative to A, and we happen to know due to previous work that A is the gene in this particular HFR strain. Do remember, different HFR strains have the F factor in different locations and different orientations. So this, you have to know what HFR strain you're dealing with in order to design this correctly. But we know that the A gene enters last, and that the B and the C genes enter earlier, but we don't know exactly what the distance is, and we don't know exactly what the order is. That's the information we're trying to find out with the experiment we're doing. So what we will do is, if we think about the F- strain, and I'm just curving the edges here, as it's not a very good drawing, but I'm curving the edges as an indication that the chromosome of the F- recipient is a circular DNA. And so what we're going to be selecting for is this crossover event. A crossover event where A+, the wild type, is being taken in and incorporated on the bacterial chromosome of the F- strain. And so that's going to be our total. We're going to select for incorporation of that one, and then we're going to say, well, we know that at least one other crossover is needed to keep this chromosome circular, right? So in how many cases did the second crossover occur between A and B, right? If this is a short distance, not very many times. If it's a large distance, probably quite a few. Or how many times was there a crossover here and then here, but not anywhere else? How many times was there a crossover here, 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 and here? That's going to be a lot less likely, right? Because there are 4 different crossover events. And you can figure out based on the genotype how many, which genes are wild type and which are still mutant, exactly where those crossovers took place. And just note, this is quite a large distance compared to, and this is quite large. We're just, we're collecting anything that had a crossover that allowed incorporation of this. There will at least be one more crossover somewhere else, and if it's here, that happens quite often because there's a big possible area here. These events in this region here between the genes that are to be mapped, they are less frequent because the distances are smaller. This isn't really drawn to scale. I probably should have drawn it to scale. That would have been a bit better. I'll try to maybe fix that in the notes before I post them for you. Okay, so that was just a sort of a rambly, quick look at, in theory, how we would do it. So this is an example. This comes out of a textbook, but this shows you pictorially and nicer than what I drew, what we're talking about. So in this case, we've got 3 different amino acid genes. They're wild type from the HFR strain, and oxytrophic mutant for all 3 from the F- strain. We are selecting for this crossover. So we know one crossover has taken place. So we know that the reason why we do that is, first of all, we have to have a reference point. And the reference point is relative to the leucine gene, which we know is last to enter. How far away are the other 2 genes and what order are they in? That's the question we're asking. And so we need to start with this, this event. And then, so if we do that, we played on media that doesn't have any leucine, but does have the other amino acids. So we can actually use, it's called a dropout powder, and you can buy it pre-made, and it has everything except leucine. There's another one you can get that has everything except methionine and so on. There's different combinations of them that you can buy, but you can also just plate them on minimal media and add arginine and methionine so that cells of all 4 types, so these are the types that we're going to get: leucine, Arg minus, methionine minus, leucine and arginine wild type, but methionine mutant. Wild type for all of them, and leucine wild type, and what mutant for arginine but wild type for methionine, all of these will grow on media that doesn't have any nutrients added to it except arginine and methionine. So that's our way of getting our total number of cells, and there's going to be lots of these guys. And then there's going to be some of these ones. And that the exact number is going to depend, of course, on the distance apart. If this is a much bigger distance than this, say, then these ones will be seen more frequently than these ones because there's more places where the crossover can occur, right? So remember, the number of crossovers is an indication of the distance between. And then there's going to be relatively few of these. And if you look at it, it's because so the wild type leucine is taken in, and then we have a crossover, arginine minus, and then we have another crossover because we've got to pick up the methionine plus from the HFR, and then methionine back to the chromosome. This last crossover is necessary to close the circle. Otherwise, remember, if we have an odd number of crossovers between a linear piece of DNA and a circular piece of DNA, the nucleases in the cell will go crazy because the chromosome is supposed to be a circle, a closed circle with no actual ends, and these nucleases just start at one end of a DNA molecule and start chewing away at it, right? So we can figure out by the relative size what the 4 crossover class is and what the various single crossover, double crossover classes. But think about this in analogy to the 3-point cross that we talked about with eukaryotes, because there was the non-crossover one, which is kind of like this one because there's no crossover between the genes we're studying, just one to either side, right? And then there's the single crossover classes here and here. There's just one crossover in the region between those genes. And then in this category, this is like the double crossover class because we've had 2 crossovers between the genes that we're testing. So if you think about that trihybrid test cross and then think about it in analogy to this, even though we're talking about 4 and 2 crossovers, really the crossover at the ends, it doesn't really count in a way because all that's doing is keeping the chromosome circular, right? And giving us basically a starting point. So hopefully that was clear. If not, it's perfectly fine. Nobody ever comes to my office hours, but you are welcome to come anytime, even if you think your question is silly or something or obvious. I'd rather be answering it because then I know, that that my office hour is actually useful for something. Okay, so let's just do a practice question. We're going to map 3 genes: methionine, thymine, which is a B vitamin, and purine, which is the precursor of nucleotides. Excuse me, oops. And so basically, we've already done an interrupted mating study, and we know the genes are not too far apart, but we also know that methionine is the one that's transferred last in that interrupted mating study. But we're not sure if purine or thymine are in the middle of the 3 of them. And so we're going to use this merozygote. Merozygote is just a temporary diploid. It's the time during which there is some linear HFR DNA inside the F- cell. And so for a short period of time, you have 2 copies of the genes. But very rapidly that DNA is degraded if crossover doesn't happen. And so we're going to be selecting for incorporation of methionine plus. Okay, so we want to select for MET plus, and then we will be asking, again, how many crossovers occurred in each of the other regions, right? And so here's our results. And I'm going to stop the video now, but I want you to, while you're waiting, not waiting, but before you start the next one, just note down how many crossovers were required to produce each of these genotypes and see if you can determine the gene order. And then we'll go over that in the next recording. Yeah, and I think that actually, I'm just thinking about things to cover. We may do 3 prep videos. Anyway, so this is your assignment if you should care to accept it. The number of crossovers required, and then given that, what the gene order is. Is this the correct order or not? And we'll pick it up in the next video.